Hello and welcome to this video on the SUPR-DSF. This is the product from Applied Photophysics and Protein Stable, and today I'm going to take you through what the SUPR-DSF can do. Now, the SUPR-DSF is a microplate based spectrofluorimeter system that is designed to do both chemical and thermal denaturation experiments in order to investigate and screen proteins for conformational stability. And so today I'll take you through the workflow of using the SUPR-DSF. We'll go from preparing the microplate, from measuring the microplate, and then I'll show you the SUPR suite software and how we're able to set up the plate designs, the measurement, and then also do some analysis on the data that we collect so we can then start to compare protein samples or formulation examples in order to rank which ones are going to optimize our protein stability. And that then gives us an idea as to which candidates we want to progress with if you're doing some kind of screening application or drug discovery type of application there. Now, the SUPR-DSF works with conventional 384 well PCR type microplates. Now, these aren't proprietary, they are commercially available from multiple retailers. The only stipulation is we use black polypropylene microplates. And that's because we're using intrinsic fluorescence with the SUPR-DSF. So we're exciting at 280 nanometers and then looking at the fluorescence between 310 and 420 nanometers. So we're looking at the tryptophan and the tyrosine fluorescence within the protein and then monitoring that fluorescence as the protein unfolds throughout the chemical or thermal denaturation experiment. And because we're using intrinsic fluorescence, that's why the polypropylene material is important, because that reduces the background and gives us the best signals possible. And we use black colours of microplates because obviously fluorescence measurements, it's all light based, so a black coloured microplate again lowers the background of our signals. But apart from that, these are just commercial microplates, nice and cheap, nice and ubiquitous, and of a standard format, so they're easily integrated within other types of systems, including liquid handling systems or robotic systems if you're thinking about automation. And today we're going to be looking at a simple globular protein lysozyme, and we'll be running a screening type application on that type of protein where we'll look at pH, we can look at salts, we can look at buffers, but today we'll just do a simple pH screen to show you the thermal denaturation experiments with the SUPR-DSF. When you prepare your microplates, we dispense our solutions into the wells of the microplate, and then once we're finished dispensing, we normally want to spin those plates down. That just evens out the meniscus within the wells of the microplate, and then we just apply a conventional qPCR type seal to the microplate. Again, these are commercially available, ubiquitous within the industry, and these are pressure activated adhesive seals, and you just simply peel off the backing, then that just gets applied onto the microplate, and then you just use a simple paddle to then apply pressure onto the microplate to seal that microplate up. And then we just remove the tabs at the sides, and then our microplate is ready for measurement. And so with the microplate prepared and ready to measure, we'll then move into the software where we'll then set up that plate design within the software and set up the experimental parameters so that we can then begin measuring the conformational stability of our proteins. And so here we are within the SUPR suite software. We're in the design screen of the software here. Here you can see a representation of the wells of the microplate. And this is where we're going to input our design parameters so we can tell the system what we want to measure. We also then have the samples list on the right hand side, and this is where we'll be able to assign names to the samples that we're allocating. And then at the top we have the thermal ramp settings, and this will allow us to set the parameters of the experiment. Now, designing a microplate is very simple within the software. We just need to click and drag across the wells that we want to select. Right click, and then we can create a new sample and we can create a new group for that sample there. And so here we then see in the samples panel that we've got our new sample there, and we can call that protein 1. And you can see that's already allocated as 3 replicates because I've selected 3 wells within the design of the microplate here. And then we've got a group header here, and this is could be our lysozyme sample, or it could be our salt conditions here. And the idea behind this is that we're able to design the microplate in such a way that we've got groupings allocated so that when we get to the analysis stage, we're then able to rank order those groupings, and you can then start to make comparisons between those types of samples. So it gives you a means to organize your data in such a way that you can then perform comparisons between different types of conditions. And so we can then also edit the file here, and we can add, delete, change our samples as we want. So then if we want to add another sample, we can click and drag across the next set of wells, create new sample, and then we can add that into this salt group that we've added here. And again, that can be protein 2. And we can continue in this fashion and create the whole plate design based on what you've got in terms of your experiment and what you've dispensed into the microplate. Well, join me in a wee moment once I've filled out the rest of the design here, and we can then get on to the measurement side of things. Okay, so you join me now that I've input all the information for this particular experiment that we're running. So as you can see, we've got a whole microplate filled with samples here. And so, as mentioned before, we're going to look at lysozyme across a whole range of different formulation conditions. So here we can see we've got our control, which is the water sample. Then we've got various different types of buffers. And as we scroll down, we've then got the background conditions for each of those there. Then we go into a ionic strength conditions here. We've got a nice pH screen that we're able to do. And this is what we'll use in the analysis in order to investigate the methods of how to analyze data. But we then also have buffer strength effects, the effects of different salts. And so we run the gambit of formulation conditions for this particular sample. One of the nice things you can do with the SUPR-DSF, because it's microplate based, is use commercial screening plates that you can buy from multiple retailers, and they come pre-made in a 96 well format. You can just transfer those formulation conditions into the 384 well plate, spike in your protein, and then that gives you a very quick and convenient way to run screening applications with the SUPR-DSF. And the nice thing with the software, once we've made a design, we can then just save this design as a template. And so if we then have future experiments we're running of the same design, we can just load that template into the software, and we don't have to go through the process of creating it again. And so once we're happy with our plate design, we then have the options on the top here to run the thermal ramp. So we can select what temperature range we want to run from. And the SUPR-DSF can go as low as 10 degrees, and it can go up to 105 degrees. We then have the ramp rate, and typical values would be 1 degree per minute, but the SUPR-DSF can go up to 10 degrees per minute. However, we'll stick to 1 degree per minute for this experiment here. We then have the return temperature. So once the thermal ramp is concluded, this is the temperature that the system returns to so that you can safely retrieve the microplate from the SUPR-DSF. The integration time is the time that the system spends measuring the fluorescence on a well of the microplate. And this is your main control for how much fluorescence count you're going to get throughout the measurement. And so typically what we do is a quick concentration ramp just to make sure that we're collecting enough fluorescence from the proteins. And for this experiment, 25 milliseconds will be more than sufficient. So once we're happy with our plate design, we've got our experimental parameter set up, and we've got all our samples inputted and placed on the microplate. We'll then go into the measure tab, and this then lets us initiate the measurement. So we click on the start button, and then we just tell the software where we want to save the data, and then we click save, and the system will then measure the microplate. So join me once the instrument's finished measuring, and we'll then start to look at the data and do some data analysis, see if we can get some TM values, and we can then start to rank order the samples and get to better understand what conditions are offering an increase in stability for the protein. You join me after the SUPR-DSF has finished its measurement, and you can see we've got our results on the software here. So we'll take a dive into the software now and we'll look at the results, do some analysis, and we can then wrap up this overview video. So let's jump into the software. So here we are in the software. You can see we've got all our measurement data here. On the left we have our tree view showing us all the different samples that we've measured. We can select each of those and have a look at what the data is for each of these samples. We then have our fluorescence plots on the right here, and we can see this very nice transition as we go from the folded state at 50 degrees here all the way up to the unfolded state at 100 degrees here, and you can see this characteristic drop of intensity as we increase temperature because we're getting more thermal energy into the system, so there's more ways for the fluorescence energy to dissipate through non-radiative means. Then on the bottom right we then look at our melt curve, our thermal denaturation curve, and being lysozyme we'd expect a two-state transition here. And we can see this here as we've got our folded plateau at the bottom here. Then we have our transition region, and then we have an unfolded plateau at the top here. And we can see that our replicates are showing a very nice agreement. So we've got a very nice set of data here. One of the advantages of the SUPR-DSF is the fact that we capture the full spectrum. And this allows us to process the data in a number of ways in order to best optimize the data that we're collecting and the thermal denaturation curves that we're generating. So at the moment we're looking at the ratio of intensities, and this is where we're taking two wavelength positions and then calculating the ratio of the intensity values that are on there. We can also use a parameter called the barycentric mean. Now the barycentric mean is a calculation that takes advantage of the whole spectrum of data. It's effectively the center of mass of the spectrum, and this can be useful because in calculating the value from the whole spectrum we can get better signal to noise, as you can see from the melt curve shown on the software here. And we can change the ratio, or we can change the values, the limits of wavelength values that we're using for this calculation as well. And finally we can just look at a single intensity value in order to judge how much that's changing throughout the experiment. But for this example we'll stick with the barycentric mean, and we'll then progress through to the analysis tab and begin the analysis process. So here we are within the analysis screen of the SuperSuite software. We are looking at our range of different pH values in this example here, and we've got a very similar layout. We've got our tree view on the left. We've now got our table of fitted values, which is empty at the moment because we haven't performed any fitting yet, but we've still got our thermal melt curve shown at the bottom here. Now to perform analysis, all we need to do is select which model we want to fit the data to. The SuperSuite software has two types of fitting model that it can use. One is the two-state model and the other is the three-state model. Now these are thermodynamic models that are derived from the actual unfolding expression of the protein, and so that allows us to not only to fit to the different plateaus that we get from our folded and unfolded transition, but it allows us to get the Tm values and the delta H values, or the Van Hoff enthalpy values. Now as mentioned, lysozyme, being a globular protein, has a two-state transition here. So we'll select our two-state formula here. We'll then initialize the parameters. We're performing a non-linear least squared fitting here, so we need to initialize the parameters so that we can get the function within that global minimum state, so we can actually get the right results here. With that we can then click the fit button, and that then fits the data, and we can see we've got a nice green line passing through the data points here, and that looks like a very good fit there. And we could then go through each of the samples and collect those fitting data here. Alternatively, what we can do is click on the group here, this pH screen group header, and this effectively selects the entire group of samples. We can then select our model. We can then initialize the parameters for all of those different samples, then fit, and that fits for all the samples within that group there. And if we then go through each of the samples here, you can see that we've got our fit line passing through the data points, and we have indeed covered the range of different pH values here. And the table is now populated with those fit parameters. We've also been able to calculate the T onset, which is the point at which we're getting 1% unfolding here, which is another useful metric for this type of experiment. Now, we have all this information here, and what we can do is immediately export the tables of fitted values. We could also export all these plots of the fitting results using these buttons here. We can also look at the fraction folded, which is a way of normalizing these melt curves so that you can compare them with each other. And this generates a very nice plot here across the different pHs. I can zoom in on the x-axis here, and we can get a look at the data, and we can see this very nice transition as we increase the pH, and we can also download that image via this button here. What we can also do, if you're wanting to process the data or look at the data out with the software, we can use the export data button, and that will give you all the fluorescence spectra as a CSV file, so you can look at that within Excel or something. We can also export the fits, which will give you all the fit information, all the fitted parameters, including the delta H values and the plateau fit values as well. And then finally, there's a report tab, which would allow you to generate a PDF of your experimental results as well. And there you have it. We've successfully performed an experiment using the SUPR-DSF. We've covered the process of preparing a microplate full of samples, applying the seal to that. We load it into the instrument. We then went over to the software, and we've designed our experiment, designed the plate, allocated samples to the wells, set our temperature ranges, our ramp rates. We then measured that microplate, and then we've been able to go into the software, fit the data, and we've then successfully managed to compare the different samples together and determine what might be the best or which samples are giving us more optimal thermal stability than other samples. I hope you've enjoyed this video. Thank you for listening. Take care.